Our lab today is about the urinary system. In this section, which is stained by H&E stain the kidney, showing the cortical area, which is characterized by the presence of renal corpuscles. This round structure indicates the region is the cortex. In addition to the renal corpuscle, we have tubular structures, which including both the proximal and distal convoluted tubules. At higher magnification, we can identify the renal corpuscle made by a central glomerular tuft and surrounded by Bowman capsule. These are sections in the proximal convoluted tubules that appear acidophilic in color, while sections in the distal convoluted tubules appear less acidophilic in color. In the medullary region, characterized by the presence of the tip of the medulla, which is the renal papilla, containing the remaining part of the loop of Henle and the collecting tubules that open toward the minor calyx, which is lined by the transitional epithelium. The renal corpuscle composed of tuft of capillaries surrounded by Bowman capsule. The parietal layer of Bowman capsule is lined by simple squamous epithelium. The corpuscle containing a vascular pole that's characterized by the presence of the macula densa, which is a part of distal convoluted tubules. In this section also we can identify section in the proximal convoluted tubules characterized by acidophilic cytoplasm of the lining cells and the apical microvilli, while the distal convoluted tubule the cells are less in height and no acidophilic cytoplasm and devoid of microvilli or brush border. In pus stain, the renal corpuscle appear as in this figure, the tuft of capillaries, the parietal layer of Bowman capsule lined by simple squamous epithelium, and this is the urinary space or Bowman space. And we have two types of tubules: the proximal convoluted tubules containing this line, purple line, which is indicate the presence of a brush border. Pus stain staining the glycocalyx of the microvilli. These are the site of the glycocalyx of the microvilli. In the distal tubule, we cannot see the purple line, which indicates the absence of the brush border in the distal convoluted tubules, while they are present in the proximal convoluted tubules. In the pus stain, also in the cortex, we can identify the renal corpuscles within the cortex, and we have extension of medullary tissue toward the cortex. This is the medullary region. It is devoid of renal corpuscle, while the cortical region containing the renal corpuscle, and we can identify extension of medullary tissue toward the cortex. These extension called the medullary rays, which are part of collecting tubules that pass toward the cortex in between the cortical structures. The ureter